Now I call upon Anusha R from course on cbbt to present her seminar.
Thank you, Dr. Good afternoon, and good afternoon, my friends. This is Anusha from cbbt 4th set. My topic is about DNA sequencing. I think you guys know about the DNA sequencing. You have heard somewhere, right? Sequencing, the word sequence. Anybody? Do you know the meaning of the word sequence? Okay. DNA sequencing. You just remember this word. The order of the nucleotides in DNA is just enough. The order. Order in the sense, we have nucleotides, or 4 nucleotides we have in this DNA. That is adenine, guanine, thymine and cytosine. Those 4 nucleotides, how it is arranged and what is the function of that, and then what is the size of that. Reading about this or studying about this is nothing but the DNA sequencing. Topics covered here is DNA sequencing, the definition and procedure, then Sanders method of DNA sequencing, then Maxam and Gilbert method. Now we'll go for the definition of DNA sequencing. DNA sequencing is the process of working out the order of the building blocks or bases in the DNA strand of the DNA. As I said, DNA sequencing is the order, right? The function of the nucleotide or the size of the nucleotide or about the nucleotide of the DNA. Steps involved in this: isolation of interested gene, isolation of vector gene, insertion of interested gene into vector DNA allowed to multiply. Then the DNA is then isolated from the bacterial cell and sent for sequencing. The isolated DNA is transferred to a plate where the sequencing reaction takes place. Plate is nothing but the test tube or some vessels which are used for the running of the reaction. Then the mixture of ingredients is added. Ingredients is nothing but the DNA polymerase, primer and other bases or free bases, or I will say in the list. Okay, free DNA bases, DNA polymerase, DNA primers and modified DNA templates. Okay, it is used for terminating the DNA, terminating the reaction. Now look at the procedure. To start the sequencing reaction, the DNA is heated to 90 or 95 degree, 90 to 95 degrees Celsius to separate the double-stranded DNA to single-stranded. Again, the temperature is lowered to 50 degrees Celsius, which enables the DNA primer to bind the plasmid DNA. Temperature is then increased to 60 degrees Celsius, where the DNA polymerase binds to primer DNA. DNA polymerase starts making a new strand of DNA by adding the unlabeled DNA bases to target DNA. Unlabeled in the sense the free bases, the free nucleotide bases. Till terminator bases is added. Terminator bases means nothing but modified DNA bases. I say in the methods how they modify the DNA to stop the reaction. Then once the terminator DNA is added, the DNA polymerase stops making the DNA, falls away from the strand. What falls away? Primer or polymerase. It falls away from the strand and terminator stops there. Again, the new strand is separated by heating the new strand to 90 degrees Celsius. The process is repeated again and again to produce lots of fragments of DNA of different lengths. The length of each fragment depends on when the terminator bases got added. added, we get the different lengths of DNA or different fragments of DNA. From first point onwards, the DNA is heated up to 90 degrees Celsius. Denaturation of the DNA happens there. Like we have double stranded DNA and it converts into single stranded by heating into 90 degrees Celsius. Again, DNA primer is added at the temperature of 50 degrees Celsius. Then DNA polymerase is added for about 60 degrees Celsius. Then DNA bases is added. Do you know why DNA bases is added for here? Anybody? Free DNA bases. See, we have single stranded DNA. We just have, this is a strand, a known template, A, P, G, C, G. This is incomplete DNA, right? This is only a single strand DNA. Those free bases is added here like a complementary. A for T, P for A, like this. These are nothing but the free DNA bases. For this free DNA bases is what added. It comes under the ingredient. Then modified DNA nucleotides or modified nucleotide bases. Modified nucleotide bases is nothing but like a probe, right? As Barka said, like a probe. In that strand, there will be some labeled radioactive phosphorus or anything else. We have that, and when it is inserted there, when the complementary DNA runs, if it is there in that, it is very easy to screen that DNA. It is very easy to know about our sequence, correct? And this is incomplete one. We have no
ಮೂವ್ ಮಾಡಿ ಇದೆ ನೋಡಿ ಕರ್ಸರ್ ಆಫ್ ಮಾಡಿ ಮೂವ್ ಮಾಡಿ ಕೆಳಗಡೆ ಕೆಳಗಡೆ ಹೋಗಿ ಕೆಳಗಡೆ ಕೆಳಗೆ 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 ಅನ್ನಲ್ಲ ಕೆಳಗೆ ಪೂರ್ತಿ ಡೌನ್ ಹೋಗಿ ಹಾಂ ಕೆಳಗಡೆ ಆಗ್ತದೆ. ನೀವು ಪಿ ಪಿ ಟಿಯಲ್ಲಿ ಮಾಡಿಲ್ವಾ ಇದು?
PPT yeah, there you can see that electrophoresis, right? What that happens is, after this, or after free DNA bases got added and after the terminator bases got added, it is inserted into gel electrophoresis and run the gel. What happens there is we get a different length of DNA strands or DNA fragments, different lengths of fragments, and we run. There is a probe there, right? We have modified nucleotide base. When we run the gel electrophoresis after screening, we come to know the color. If we have the fluorescence here, we come to know about our sequence. It is very easy to identify our sequence. In this diagram, we will come to see that in this denaturation is taking place. Then make multiple copies of segment. ಇದನ್ನ ತಿರ್ಗಾ ಡಿನೇಚರ್ ಮಾಡ್ತೀವಿ. ಆಲ್ರೆಡಿ ಡಿನೇಚರ್ ಆಗಿದೆ, ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಇರೋದು ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಮಾಡ್ತೀವಿ. ದೆನ್ ಪೀಸಸ್ ಆಗುತ್ತೆ. ಓಕೆ, ದೆನ್ ಅಟ್ಯಾಚ್ ಅ ಪ್ರೈಮರ್ ಇಸ್ added there by increasing the temperature, by decreasing the temperature from 90 degrees Celsius to 50 degrees Celsius. Primer is got attached. This is for Sanders reagent. Okay, this is for Sanders reagent. We neglect that and come here. The elongation is taking place here. We have this is our known template DNA. Okay, And this is the new strand of DNA we have got after primer and polymer is got attached and elongation takes place. This is a new strand, and then again denaturation of bronze chains. Do you know why this is happening here? This strand is there. We'll be not knowing about the sequence. We don't know about this sequence. This is template strand. It is a complementary strand. By the help of complementary strand, we will know the sequence. Now, complementary strand and okay? We'll be having some complementary strands here. Each strand and follow, we going to find this the main template of the strand. It just detects that where we have used this probe like that. Coming to Sanger's method or enzymatic method. Sanger's method. Frederick Sanger. It was invented in the year 1977. It is also called as enzymatic method. The word enzymatic, it refers as a enzyme is used here to identify the sequence. It is also called as dideoxy method of sequencing. Dideoxy method. Do anybody know what is dideoxy method? Dideoxy. What is di?
2.
Di is 2. D. Sorry, neglect the D. Oxy. What is
Oxygen.
oxygen? Yeah. What is D?
Removing.
Deletion.
Deletion.
Dideoxy method. Deletion of 2 oxygen is declared here. In what? In a pentose sugar. This is a pentose sugar. Okay. 1 prime, 2 prime, 3 prime. Dideoxy represents removing of this oxygen in 3 prime and 2 prime. Okay. Di is 2 oxygen gets removed. D, deletion of oxygen. Oxygen is removed here. If oxygen is removed here, what will happen here? Can anybody know about this? What will happen if oxygen is removed here?
Hydrogen is present. Hydrogen will be present. Hydrogen will remain there.
Any other?
Hydroxyl.
What will happen in the 3 prime? What is attached to 3 prime?
Hydroxyl and hydrogen.
Phosphodiester bond. If there is no oxygen here, how can phosphodiester bond attach here? Right? By this only. See, if there is no oxygen here, phosphodiester bond cannot attach. Then what happens? It terminates here. It's simple. This is what's called as a Sanger's method. He used this method. Okay? So you can look at it. DDNP is nothing but dideoxy nucleotide, and deoxy is a normal nucleotide, and this is dideoxy, and oxygen is removed, 2 oxygen gets removed, and the chain terminates. Procedure for that. The unknown sequence is taken for example 3 prime A C C G P A C. This is an unknown sequence. For our reference, we have a name for that. A C C G P A and C. 4 different vessels are used by Sanger. As he uses 4 vessels. In each vessels, he introduces a normal ATP, GTP, CTP and TTP. He puts unknown DNA sequence in different vessels and BD is nothing but dideoxy. BD-ATP, BD-GTP, BD-CTP and BD-ATP is added respectively. Then one of them is removed. This in the sense this is one of the vessel. Okay. What we are added? There is a BATP. We have normal DATP, DGTP, DCTP and DTTP. And if we add DDATP, then this gets removed. Now what we have in this result? DDATP, then DGTP, DCTP, DTTP. Okay?
ಅಲ್ಲಿ ನಿಂತ್ಕೊಂಡೆ ಮಾನಿಟರ್ ಮಾಡಿ
If the sequence is like that, A, C, C, G, T, A and C. We have A here. This is a complementary for that strand. It is in the fifth place, right? A, C, C, G, T, A, C. It is in the fifth place, right? D. A complementary for A and C. It is in the fifth place. When this got added, up to here the reaction runs, and after while coming to this T, here it gets terminated. This is how we get the different strands of DNA. Okay, this is very important. We have there should be different strands of DNA of different lengths of fragments. Then all the fragments are taken and inserted into electrophoresis and run the gel from negative to positive. After this, we get some different lengths of fragment, right? After that, we have to insert them into electrophoresis and have to run the gel. Now we can read the sequence from smaller one to larger one. We all know that for smaller DNA, because it has a very less number of nucleotides, a molecular weight, based on molecular weight, it arranges from smaller one to larger one. Smaller one, it goes faster, okay, towards the positive end. It runs faster, and then the last one, larger one will come. The sequence we got here for the sequencing is complementary gene. As I said, the sequence, this is a unknown sequence, right? Unknown sequence, or it can be read as a template strand. And what we got after this, after the electrophoresis, what we got is nothing but as a complementary strand. Okay, complementary gene or complementary strand. For that, we have to write our template strand, and that is our sequence. Okay. See this diagram, you will come to know what is happening in this Sanger method. Single-stranded DNA with the unknown sequence serves as a template, plus DNA polymerase. As I said, DNA primer is used, and then DNA polymerase, it runs and elongates, and DATP, DCTP, DTTP and DGTP is got added for that radioactively labeled primer. Or the DDTP, DDATP, DDCTP, DDPTP and DDGTP, deoxy-ATP, CTP are got added there. diagram, we have different lengths of the fragments, right? DDA, we have only for the adenine, we have got 3 fragments here. Longer fragments, shorter fragments, and this is the electrophoresis B sequence. After this, we have got some different lengths of fragments, right? Then we'll get this sequence, this complementary sequence. For that, we have written the template structure. Okay, this is the end of the Sanger's method. Advantages of that: it is simple and cost effective. It is simple and cost effective. Best when applied to a short sequence. We can't take long sequence. A short sequence it also applies for the advantages and also disadvantages, because it cannot be applied for the human genome. It is a long base pair, right? We have so many base pairs in that. Only for the short sequence we can apply Sanger's method. Disadvantage: cannot be applied for a long sequence like human genome. Then Maxim and Gilbert method of DNA sequencing, also known as chemical degradation method or chemical sequencing. Chemical degradation. Okay. Keep it in that mind. Keep this in mind. What is the chemical? We are using chemical here. For Sanger's method, what we have used? For Sanger's method, what we have used?
Enzymes.
Enzyme. Here, chemical. You have to make some differences between those two, because we have this for our in a part of examination. It's very easy to remember. It was invented by Alan Maxim and Walter Gilbert in the year 1977. It is a method by which sequence of a DNA identified by using chemicals. Chemicals is nothing but radioactive phosphorus, iodine, and sulfur. Those are used here. That cuts the DNA at specific points. Procedure. The unknown fragment of double-stranded DNA is taken. Here also the unknown strand is taken. End labeling is done first. End labeling is 5' or 3', like end labeling. Removal of normal phosphate by alkali phosphatase. What is that enzyme? Alkali phosphatase and gets dephosphorylated. Removal of normal phosphate. We have phosphate in this, right? We'll be having phosphate.
ಅಯ್ಯೋ ಕಟ್ ಮಾಡ್ಬಿಡ್ತೀವಿ ಇನ್ನೊಂದ್ಸರಿ ಹತ್ರ ಹೇಳಿಸ್ಕೊಂಡ್ರಿ ಅಂದರೆ ಸೆನ್ಸ್ಲೆಸ್ ಕ್ರೀಚರ್ಸ್